So I'm excited this morning to introduce two exceptional individuals who are absolute paradigms of what we've just been talking about. People who are doing phenomenal basic science, but with their eye on where it can take the field and move us clinically. And I would like to first introduce Dr. Irv Weissman, who is a professor of pathology and developmental biology at Stanford University, where he also serves as the director of the Stanford Institute for Stem Cell Biology and Regenerative Medicine. Irv received his MD from the Stanford School of Medicine, and his research encompasses a very broad swath, but is particularly focused on the phylology and developmental biology of cells that make up our blood-forming and immune systems. His laboratory identified stem cells in the blood lineage of mice and has gone on using lineage analysis to show how those basic stem cells move into mature cells of the blood-forming system and the immune system in mice. He's also done similar work now in isolating human stem cells from the blood, from the neuronal system, and more recently from patients with human myeloid leukemia. In addition, the Weissman Laboratory has pioneered the study of genes and proteins that are involved in cell adhesion events required for lymphocyte homing in normal and diseased states. He was a founding member of the Amgen, DNAX, and T-Cell Sciences boards and has received the Kovalenko Medal of the National Academy of Sciences, the de Villiers Award of the Leukemia Society of America, and the 2002 California Scientist of the Year Award from the California Science Center. We're honored to have Dr. Weissman present to us this morning.
Stem cell biology is a new field. It's a field unto itself because it deals with not only the genesis of organs during development, their maintenance during adulthood, and they really are the core of this field called regenerative medicine. There's no other field that had that as a discipline, and so naming it as a discipline and funding it here is incredibly important. My own research on, and the examples I'm going to present you, was really about trying to figure out how the thymus made immune cells. And in a search for the cell that went from the bone marrow to the thymus to become T lymphocytes, we had an observation in the lab that led us to say, certainly led me to say, what we're really looking for is the stem cell. We're not looking for just a thymic progenitor. And this goes on over and over in basic science. If you keep your mind open, you will start to see things. Now, stem cells are defined as cells which at the single cell level, when they divide, can both self-renew and give rise to daughter cells that are committed to differentiate. In the blood-forming system, blood-forming stem cells are 1 in 20,000 cells in the bone marrow, but they are the only cells that when they divide make another copy of themselves by a process called self-renewal, which utilizes many gene programs we now know to keep self-renewal going, but to keep it in check so that you don't have unregulated self-renewal, which is the central problem in cancer. So we had a search, a basic science search, not only for the T lymphocyte progenitor, but for blood-forming stem cells next. And we found first in mouse and then in humans molecules that are on the stem cells and a large number of molecules that are not. And we had to find a way about knowing which molecules were on and which were not to purify those cells to homogeneity. Next slide. So we were lucky that at Stanford, the Herzenbergs had developed a fluorescence activated cell sorter, which was the result of an initiative that Josh Lederberg had in trying to understand the physics and engineering of many processes. He was interested in the Mars landing, but it brought people in conjunction to develop eventually the fluorescence activated cell sorter. So we used the cell sorter to separate from the population from blood-forming organs on the left, if you can see it. HSC stands for blood-forming stem cell. I'm sorry for the Latin. It's hematopoietic, but it's a blood-forming stem cell. It's the rare cell there, and we want to get it pure over on the right down. We want to get it pure because in patients who have cancer that is spread to the bone marrow and the blood, we want to be able to give them treatments that would kill them while you're trying to kill the cancer cells, and then you regenerate their blood-forming system, you hope with pure stem cells, not with cancer-contaminated stem cells. We also want to transplant stem cells from one individual to another, where the problem is the preformed immune system of the donor, when transplanted into a host, can make an immune response that's worse than the disease they have. So we wanted a common method for getting out pure stem cells, and we did. That was the previous slide. Next. So this is a clinical trial that was done by a company that I co-founded. I had stock in and got consulting fees from, but it's gone now. It was sold to Novartis, sort of against my will, but it was sold. And right in the middle of a trial, we had isolated from women with metastatic breast cancer, so it had spread throughout the body. All of them had macroscopic cancer outside of the breast, outside of the lymph nodes. Rob Nagin in the group isolated pure hematopoietic stem cells and reduced the frequency of transplants that were going on that were contaminated with cancer cells from 40% down to 0. Nobody got cancer cells back within the limits of what we could look at. And what you're looking at is the end of the clinical trial, which finished essentially after Novartis bought the company. In pink is overall survival. In blue or green, whatever you're seeing, blue, is the cancer-free survival, and that's the fraction of patients. What you can see is that 2 and 3 years, it was 45% in a phase 1 trial of alive without disease. The little x, it's not a legitimate case control, was the results of a trial by Statmauer in Pennsylvania of the East Coast Oncology Group, where instead of purifying the stem cells away from the cancer cells, they gave everything back to the patient after essentially the same kind of therapy. So even I can see that 6% is different than 45%. It should have been enough in the early 2000s to go to a clinical trial, but then you get into business issues. And when the patent runs out, and what trials you can do to validate this, and I'll just say Novartis decided not to follow it up. But this is the promise of regenerative medicine. If you had liver stem cells, you could treat the liver for metastasis. If you had lung stem cells, I predict you could treat the lung as well. Next slide. Now, this is a donor to a host on the left. If you give the full population of cells that include T lymphocytes as well as blood-forming stem cells into an irradiated host who has, let's say, a leukemia or a genetic disease of the blood-forming system, the pre-existing T lymphocytes that the donor had rejects every tissue of the host, and what the clinician has to do is to treat the graft versus host disease. Now, our objective was to be able to give stem cells and get the regenerative part without having graft versus host disease. And I hate to say this now, it's 17 years ago that we, Judy Shizuru and I, thank you. Oh yes, wrong. Is that showing anything for you? I can't see anything. Oh, the red button. Well, anyway, it just doesn't show very well. So just trust me about many, many things. We found in mice, and in mice that had human blood-forming organs, that when we isolated the stem cell, we could regenerate the organ and no graft versus host disease. Now, we wanted to be able to apply that to a whole host of diseases that are not just malignant cancer diseases, but regenerative. If you're born with sickle cell anemia or thalassemia or severe combined immunodeficiency or any, essentially, of the genetic autoimmune diseases, this is what you want to be able to do. So pure stem cells. Now what I said there is that the host has a donor blood-forming and immune system. And what we've known for years is that if you have, in fact this is an experiment I did in high school, if you have a donor blood-forming and immune system, when you transplant from the CFT7 black mouse to the BAL mouse stem cells to make a blood-forming and immune system, it cannot reject itself. So you can do at the same day or any time a heart transplant, a skin transplant, or a pancreatic islet transplant. And if it comes from the same donor, you need no immunosuppression. You just need to have the stem cells that become established. So that would be a fantastic advance. We've had this for about 18 years, and it has not moved into clinical practice. Next slide. So you know that, of course, this society was founded on the basis that the government was not going to fund certain kinds of embryonic stem cell research or certain kinds of embryonic stem cell lines. I want to remind you that when you have an embryonic or pluripotent stem cell line, whether it's IPS or nuclear transfer or straight established from a blastocyst, that it, in order to make blood, has to go through a blood-forming stem cell. In order to make brain, it has to go through a brain-forming stem cell. So now that we've established that a blood-forming stem cell can induce permanent transplantation tolerance to any other tissue or organ from the same donor, I think that is the platform for what we are doing. If we can establish viable, transplantable, hematopoietic or blood-forming stem cells from embryonic stem cells, we open up the whole field of replacing the blood-forming and immune system and co-transplanting or transplanting later the tissues, organs, or tissue stem cells that are necessary. This is not science fiction. We have done it over and over again in the mouse models. Next slide. So autoimmune diseases are a peculiar blood disease. Everybody thought in the beginning that the disease type 1 diabetes, juvenile diabetes, was a problem of the insulin-producing cells, but it's a problem of the immune system. And that problem of the immune system is that you make T cells because of the genetic defects that attack and kill your insulin-producing cells. So I'll just do this one fast because probably my time is getting close, is that you're seeing on this graph the time when mice get type 1 diabetes because they have the same kinds of genetic defects that children do who get type 1 diabetes. If you transplant their own bone marrow back into them after you condition them by removing their lymphocytes with antibodies and you irradiate them, bring them next to death, you gain about a month, and that month you gained is exactly what Richard Burt is doing at Northwestern. If you do pure stem cells, so you're not transferring the T cells that cause the disease, you gain another month. But if you transplant at the time of polydipsia, polyuria, drinking too much, peeing too much, so they still have some islet left. If you transplant blood-forming stem cells from a disease-resistant donor, you block the progression to full diabetes. Chou and I did that, and we also have gone on to show an already diabetic mouse can be cured with a co-transplant of blood-forming stem cells and islet cells from the same donor. So this is basic science that has moved into a very practical outcome. So you could ask, we did the same thing for lupus. You could ask, why isn't this a therapy? Well, if you're a child getting insulin, and you may have a shortened lifespan and lots of bad diseases, that's better than dying of the radiation or the chemotherapy. So the next objective is, how can you change from chemotherapy and radiation, which were the tools of the oncologist who invented bone marrow transplants. They did it for the reason they wanted to go to a higher and higher dose. So we wanted to replace the irradiation which kills all dividing cells to using antibodies selectively to remove the immune cells and eventually the blood-forming stem cells. Anishka Chekowitz, a medical student at Stanford, and I found that one antibody in mice can remove the mouse blood-forming stem cells with hardly any other damage. And so if you transplant, you go from transplanting immunodeficient mice with stem cells alone but no conditioning, that's the white circles, to pre-treating with one dose of the antibody that eliminates stem cells and follow that up. Now you have on the order of 15% chimera. And if you just do that 3 times in a row, antibody deletion stem cells, wait a week, antibody deletion stem cells, wait a week, antibody deletion stem cells, it's now 90% donor-derived cells. We plan to go forward with this to treat immunodeficient patients, we hope with Mort Cowan here at UCSF, Judy Shizuru and I. But this is the example. We have found now an anti-human antibody against the same molecule that eliminates human blood-forming stem cells. So that will move its way toward trials, but it all comes from that basic science. If I have time, I want to say one more thing, which will take a few minutes, but it's, I think, really unexpected and important. Don't look at that diagram for a minute. That diagram shows across the top each step of the blood-forming system to go from a blood-forming stem cell on the left to the blood cells on the right. The one thing to remember is the only self-renewing cell in the blood-forming system is the blood-forming stem cell. It is the only one that can accumulate the damage that leads to cancer, in this case, myelogenous leukemia. In a mouse experiment in 1997, David Traver, now on the faculty at UCSD, and I put in 2 genes, that's number 1 and number 2 up there, that simply blocked programmed cell death in the blood-forming system. And surprisingly, 30% of the animals got acute myelogenous leukemia by 8 weeks of age. So all we did was we blocked programmed cell death. When we got out the leukemia cells and making good guesses, we found at least 5 other genetic or epigenetic inherited changes in gene expression had occurred. And in every mouse, the same ones had occurred. So that said, first, that you have a long progression, that's down the left, in the self-renewing pool of building up the pre-leukemic steps. But then when you get to leukemia, we found that the leukemia in mice was transferred with a cell called a progenitor, GMP, and we later showed in humans the human leukemia almost always resides in a multipotent progenitor that can't self-renew. We can transfer the disease from a patient into an immune-deficient mouse with those cells and only those cells. That subset of cells in the leukemia that represent 5% of the cells in those particular leukemias. Along the way, look at number 5. In looking at the mouse in maybe one of the first microarrays that we did with Affymetrix, 1997, 1998, the gene CD47 popped up. So all the rest of the story is following that chance observance, that leukemia stem cells express CD47, a cell surface molecule, and that normal stem cells don't. CD47 is a molecule on the surface of aging cells, on young cells, not aging cells, that prevents them from being eaten by the reutilization system of macrophages. It's a don't eat me signal. Thanks to Eric Brown, formerly here, now at Genentech, and others, this signal is understood to interact with a receptor on macrophages to say don't eat me, and the biochemical pathway is known. Well, that was shocking. The mouse leukemias had developed a don't eat me signal, and obviously if the leukemia is protected from macrophages, maybe that plays a role in the pathogenesis. So Ashalizadeh, an MD Ph.D. from Stanford, looked at all of the published microarrays of gene expression in the human myelogenous leukemias. And what that shows is that for every subtype, you have an average of 2 to the 9th fold increased expression in CD47 versus normal cells. Every subtype of AML. So it went from a couple mice to every acute myelogenous leukemia in humans. If you now, as Ash did, look at the level of CD47 in published patient samples along with their outcome, those that were CD47 high were all dead before 5 years, no matter what the therapy. Those were in the lowest 40% of CD47 expression, 45% alive at 7 years. Obviously an independent risk factor for the disease. So we did the straightforward experiment and showed that if a cell has CD47 on its surface, on the right, on the left, it is not eaten by macrophages in a test tube. If it doesn't have CD47 on the right, all of those cells are inside and being killed by macrophages after they're eaten. So Mark Chou and Ravi Majetti, Ravi is a medical school graduate, MD, Ph.D. graduate of this institution, but now he's at our institution and we hired him. We treated primary human acute myelogenous leukemia, transplanting its leukemia stem cells into immunodeficient mice. On the top, you see the bone marrow is full of leukemia. On the bottom is after 3 weeks of therapy with anti-CD47 alone. That was curative. Now, I don't believe for a minute that a single antibody is going to cure leukemia because we know from the genetic experience of Lurie and Delbruck and Letterberg that you always have variants. And if the variants aren't recognized in CD47, are not recognized by the antibody, they'll grow up. And that is essentially the story of rituximab and Herceptin, single antibody monotherapies. So we want to carry this much further. We want to do 2 antibody therapies and prove whether there's synergism or not. And if there's synergism, we want to have help at the FDA to say, well, here is a case where it's immoral to do monotherapy because you may make a lot of money, but you're not going to cure patients. Are you with me still? When we looked at the expression of CD47, it wasn't limited to leukemias. It's on human bladder cancer, human ovarian cancer, and on the bladder cancer, it's on the cancer stem cells. That's what this slide shows. And Keith Chan and I have now shown that antibody therapy of a primary bladder cancer in the skin of a mouse, anti-CD47 shrinks the tumor. Now, we haven't optimized this at all. But the point I'm trying to make is the chance observation, when we tried to look at the genesis of a mouse leukemia, came up with a marker. That's not hypothesis-based science, that's observation. And then you have a hypothesis, and the hypothesis is, if you have a don't eat me signal, it may be important, and it is. Thank you.